to Lab 6. Can you actually hear me? I also have a microphone. So are you okay today with my voice? Sometimes when I get tired, it goes soft. Okay. So Lab 6 is really is to purify the red fluorescent protein. And not every teacher will do this, depending on how much time you have. But it's a very, very interesting lab. So in this great big picture that we have, what we've done, here we go. Okay, so remember we've done all of this where we've actually done the restriction digest, made the recombinant plasmid, then you verify that you had the recombinant plasmid, and then you're doing the transformation. Okay, so now that the transformation has happened, you're hoping that the bacteria cells are going to start producing, dividing more and more and more, and they're also going to replicate the recombinant plasmid that they have inside of them. And under the right conditions, they will start expressing that protein of interest, the red fluorescent protein, okay? And so we need to purify that protein. Okay, so why do we do this? Is really to try to show students what a biopharmaceutical company does, right? The whole point of these pharmaceutical companies is to produce lots and lots of pure biological medicines, okay? It's called the biologics. And in this case, I think we all started with insulin as our example, right? Mm-hmm. For this workshop. Okay, yes. And so we want to be able to purify insulin, purified so clean that they can be injected into a patient, right? And so that's the problem. You're asking bacteria cells to make this protein for you, but they of course make thousands of other proteins that they need for their own cells, and you need to get this one protein out, this one human protein, or in this case our coral protein out. Okay, so how do we isolate this? Is we're going to use an expression vector. So it's a very special type of plasmid, so that the cell is going to be making a large quantity of this one interesting gene. And so in this case, the red fluorescent protein gene, and then we're going to make a whole bunch of this red fluorescent protein. Okay, so I don't know if you all did this in molecular biology in college, but I italicize, our genes are italicized, and then the proteins are in caps. Okay, so you know the difference.
Say that one more time.
So gene names are in italics, mm-hmm. and then the protein names, if you're doing acronyms, are going to be caps. Okay, so just so you know why I wrote this. Okay, and the way that we're going to purify this protein out of the huge gamish of bacterial and other proteins is we're going to use column chromatography. And if you remember way back from college chemistry, there's lots and lots of different chromatography methods to separate, different mixtures. And in this case, we're going to try to understand one very specific thing about the protein. We understand the amino acid makeup of this red fluorescent protein, and has a very specific characteristics, unlike a lot of other proteins, and that is it's very, very hydrophobic. Okay, there are many parts of this protein that is hydrophobic. Right. So anyway, so that's what we're going to do in 6B. So Lab 6 is actually got two compartments. One is 6A, which is really to take your culture of bacteria cells and you're going to lyse them open. And so you're going to get all of this, all the cytoplasm from these bacteria cells in a tube. Okay, and 6B is going to actually be purifying the actual protein using column chromatography. So for each of you, you're going to need to decide how much of this you want to do. Okay, I have a lot of teachers who totally skip 6A. Okay, and they just come to me, and all we give them is the supernatant, and all we do is column chromatography. So maybe in the beginning, that's what I would suggest. Okay, if you're going to start, and this is your first year or second year, maybe just do 6B. Okay, but in this workshop, we want to teach you the whole thing, so at least see it once. Right. Okay. So before lab 6, we're growing this large bacteria culture. So Karen showed you yesterday how to inoculate the flask with your lab 6 vials, and then you get the pink. Did you already take out the culture?
Yeah, it's right here.
Oh, okay, sorry. All right. Okay, so so here's your Betty Crocker. Okay, so this is the lab 6 cells culture, liquid culture. So this liquid culture has millions, maybe billions of cells, millions probably, millions and millions of cells in there. And you can tell that there's cells in there because when you try to look through it, it's not transparent, right? Mm-hmm. It's turbid, it's opaque, okay? And, but the cells were grown in media, right? And media, liquid media is the nutrient broth. You don't want that to be in your tubes. That's just like excess food, and so you want to get rid of that, okay? So as part of lab 6, you need to get rid of the broth, and you need to concentrate the bacteria cells into a pellet. And then you take your bacteria cells and you have to lyse them open. And so we're going to use detergents and enzymes to try to do that. Okay, so that's what lab 6A means, is to lyse the cells. Lab 6B is then to actually do the purification using the chromatography that I mentioned. All right, so before lunch today, we're going to do lab 6A, go through the steps of that. And okay, so here, sorry, let me see my slides. Okay, so I'm doing this in the test tube just to kind of give you the idea. So overnight culture is now totally pink, right? And then what we're going to do is we're going to centrifuge, and at the end we're going to see the cell pellet. Okay, and can you see on here? Where is the pink? Is it in the top or the bottom? At the bottom. And so what's at the liquid here? That's going to be the broth, okay, that we're trying to get rid of. Okay, and so the next thing we're going to do is we're going to throw away the broth, and then we're going to add detergent, okay? And when we add the detergent, it breaks open all of the cell membranes, and then all of the cells have lysed open, and now all of the liquid in here will be the lysis buffer with all this cytoplasm. Does that make sense? Mm-hmm. Okay. And then we're going to spin it down again. And now look, where's the pink now? Can you see it? It's on top. It's on the top. So that's what we call the supernatant. And the pellet now is all the cell debris. All the cell walls of the bacteria cells are now the debris. Okay? So what do you want to throw away? The debris. And what do you want to keep? The pellet. Supernatant. Okay, so don't mess those two up. Okay, so in this one, in this first step, we throw away the broth, and the next pellet we have, the next two that we centrifuge, we want to keep the pink supernatant. Okay, so just follow the pink, right? We're lucky because the protein that we're trying to isolate is pink. It's fluorescent pink, and most proteins are not colored at all, but we happen to choose one that is colored, so it's easy for students to follow. Okay, so then what we do is we're going to add the protein with a very high salt, and then we're going to run the chromatography, and then it'll come down and we'll purify that pink fluorescent protein. Okay, the red fluorescent protein. Okay, that's the overall idea. Does that make sense? Mm-hmm. So lab 6A is just this part right here. We're going to do this part. Okay. All right, so not much talking. Anyway, the reagents that we're going to give you is E. coli culture. So as a teacher, I typically will have aliquoted those ahead of time. Do we give them EC? Yeah. So you see at your table, you should have a tube of pink EC. So are these whole cells that came from here, or are they lyse cells? Whole cells. Whole cells. Okay, so that means they have a potential to hurt, don't let the kids drink whatever you eat in here.
These are for the protein purification and not the lysis?
Yeah, so before lunch we're just going to do the lysis, and after lunch we'll do the chromatography columns, right? Okay, and then you also need to have, it's called the elution buffer, okay, and then you need to have lysis buffer, Okay, and so I'm just going to show you, lysis buffer is basically a detergent, and it's got an enzyme called DNase. What do you think that means? What does that do? ASE stands for enzyme that breaks down the DNA. That's right. And lysozyme, that's the enzyme that's in your tears, and it breaks down the cell, the bacteria cells, okay, specifically for bacteria. Okay, and so basically we're trying to break down the bacteria cell wall. We're also trying to break down the bacteria genomic DNA. So it's in little chunks, so it'll be easier to get to it. Okay, and then students will, so at your table, you will now have waste beakers. So again, I call these my bio-waste beakers because I want students to understand this is still biohazard and that we're going to, you can't dump it down the sink, you are going to put it, and we'll put it into the autoclave later. You should have P1000s now, the large pipettes and the large pipette tips. Do we all have that?
I'll get those out, I'll get them out.
Okay. And then you're going to have centrifuge tubes, okay? And so now, are we using high-speed centrifuge or we're going to still use the blue ones?
There's a high speed that put it out, we're gonna go, yeah, right there.
Okay, all right, so now we're going to use a high speed. When we say high speed, I'm just telling you, these are about 5,000 RPMs. This one goes to about 14,000 RPMs, so that's what we call by high speed. And it's a variable so that it will actually change the speed.
For the 1,000, yeah, they should be blue usually.
Okay, and then you're going to have autoclave bags, again, because we're going to collect your biohazard later.
So
Okay.
you're gonna need different tips because it's a
Yes, so you will need the blue or the larger tips.
Okay.
They used to be blue, so that would be.
So for this lab, you only really need a P1000.
Yeah.
The last lab was the only lab you need more than one. So if I were you, I'd put away the P20s and the P200s. Students don't get the rate.
And also take away the tip box at the same time.
Yeah.
So they, okay. Confused. All right, so you all have the flow chart for this lab. Okay, and we just wanted to make sure, in the old edition of the student guide, there was a mistake in the picture, and it said to flip the tube upside down. So we don't want you to do that because you might contaminate the dust, okay? And we also want you to be very, very careful. Well, we're not going to quite do this yet, but when you get to the final cell pellet and you're removing the supernatant, don't mix it, right? Try to make sure when you centrifuge, the pellet stays a pellet and the broth is at the top. So my rule of thumb in my class is if you're coming to a centrifuge, you must bring your tube rack with you. And you put your tubes into that rack and you bring it back this way. I feel like when students carry them by hand, they tend to jiggle a little bit more. So, okay, that's my rule. Okay, any questions on this? So we're just going to follow the directions. In this case, you're basically going to be putting. You're going to centrifuge the EC tubes for 5 minutes, okay? And in your tube right now, there's only 1 mil. Can you all see? Can you look in there? There's only about 1 mil. And that's okay, but it would be better if you had more cells because then you would have more product at the end. So it'd be more pink to do your color chromatography. So this is what we do. I typically will actually have the tube set up so that the students come in, I've already centrifuged for them, but anyway, and then we add, after they centrifuge, dump out the broth, we add another 1,000 microliters or 1 more mil of this broth to the tubes. So then you spin it down, so you really have a total of 2 mils worth of culture cells in your tube at the end before you start lysing. Did that make sense, what I just said? Any questions?
But today we're just doing the lab.
No, we're going to do 2. Because
we're
we
gonna
want you, you to be the students. We want you to see how the protocol is following through. Okay. Anybody? Questions? Okay.
Yeah.